like to illustrate here the mammalian imprinting cycle. And I'm beginning here with a zygote who's inherited one chromosome from a sperm and another chromosome from an oocyte or egg. And for the purposes of this example, we're going to say that this maternal chromosome, the chromosome that came from the oocyte, is methylated. So some gene or region has been marked with methylation. Now what we know is this zygote is going to divide many, many times to give us a person. If this is a human, could be a mouse or a pig or another kind of mammal. And this happens through mitosis. And so in the somatic cells, every time this cell divides, it will replicate its DNA and restore the same marks. So in this example, say that this gene is expressed in the brain. Actually, only the paternal copy of that gene is being expressed. So we're seeing monoallelic expression. That other chromosome has been silenced by that methylation. And so this is the stage that we would call maintenance. So mitosis is maintaining the marks passed from the gametes. But in the primordial, so I'm going to draw this here, primordial germ cells, we are going to see that the marks are erased. And so now we're going to have two chromosomes that are free from marks. And as these undergo oogenesis, so producing, we're going to assume that this individual has ovaries. It would simply be the opposite pattern if this was a sperm-producing individual. And so now what we're going to see is every oocyte also goes through the establishment phase. So we had maintenance, we had erasure, and now we're re-establishing this mark. So every oocyte this individual produces will have a maternal methylation mark, regardless of where that chromosome actually originated. So a female or an ovary can only pass on ovary imprinted genes. Okay, and so then this cycle would just begin again if this oocyte becomes fertilized by a sperm from another individual. So these are the three steps: maintenance, erasure, and establishment.